I look a little white, but other than that, can you guys confirm that you can hear me?
I can hear you.
Thank you. Let me call up my lesson. All right. So today is June 4th, and we are supposed to be covering the start of chapter 13. Let me share my screen. Come on. All right, you should be seeing my screen now. There is no lab today. I will be logging in for 15 minutes to answer any questions. After 15 minutes, I'll log out. Any question about any of that?
I do have one question, or more of a clarification.
Mhm.
I just want to make sure we don't have to do anything with the quiz 5C that's due, you have due Thursday. That's more of a point system for you.
I'm sorry, I didn't quite understand.
The quiz 5C assignment, we're not supposed to do anything with it, right? That's just for you.
Quiz 5C.
Yeah.
Yeah, there's a quiz that I'm going to be changing the amount of the points, meaning it was worth something like, I think it was worth 0.2%, or no, 0.2 points. And I'm going to change that so it'll be worth 75 points. And that score will be in the quiz 5C column.
Okay, because we have just an assignment that we can see right now that's labeled as a second quiz 5C. So I just make sure that we don't have to actually do anything
You
for
have
it.
an assignment?
Yeah, it's listing right due Thursday. It's 0 points, but it has an assignment listed for quiz 5C. And you have a whole description describing the whole point system and how it's complicated. So I assume.
Okay. Yeah, so the description should tell you you don't need to do anything.
Okay, thank you.
All right. I didn't realize that was an assignment, but I guess it has to be, because if it's not an assignment, I can't make it worth points. And this is to make your quiz 5C worth 75 points, but I can't put it in there as explained on the sheet, because if I did, then the total number of quiz points would be too much. And your quiz points will be the points that are detailed in the syllabus, and you can throw out one quiz, but I still have to make the quiz worth 75 points. And so this is how I'm doing it. All right, let's move on to I think we need to finish up chapter 14, but maybe we started chapter 13. I don't remember. My voice is kind of hoarse. I'm not sure why. So I'm going to take a cough drop and hopefully that'll get better. If you are bothered by my talking with a cough drop, I'm sorry, but my voice is for some reason very hoarse today. I don't have a sore throat or anything, but it might be just the changing weather. Oh, it looks like we did finish chapter 14. Good. So we're on chapter 13. So last time we talked about viral structure, and mentioned that viruses have a genome which is nucleic acid, and it can be DNA or RNA, never both. And it can be double-stranded or single-stranded. Double-stranded RNA is very unusual in our cells. Our cells do not have double-stranded RNA, but some viral genomes are double-stranded RNA. And then if it's a single-strand RNA virus, you need to state whether it's the positive single strand or the negative single strand. So all viruses have a genome, and all viruses have a capsid. That is a protein coat surrounding the genome. The capsid, let me see if I can blow this up. The capsid is composed of capsomeres. And so this structure here is the capsid with this side cut out. And each one of these little round things is a capsomere. Sometimes, like in this virus, there's only one capsomere and it just repeats to make the entire capsid. But with other viruses, there are different proteins which make the virus. And then on some viruses, not all viruses, but some viruses, they have an envelope, which is a lipid bilayer surrounding the capsid. Here we can see the genome right there, and this is the capsid right there, and around it is the envelope. The lipid bilayer does have proteins and carbohydrates in it, but it is mainly phospholipid, actually. Only some viruses have an envelope. We call these envelope viruses. A virus without an envelope is called a naked virus. External to the capsid, not the capsid, external to the envelope, and actually inside the envelope are spikes. They're usually glycoproteins and protruding outside the envelope. These spikes, if the virus has them, they will be the antigens protruding from the envelope surface. An antigen is what a host, like people's immune system, recognizes as foreign. And what we will recognize as foreign will be the outermost molecules. In the case of the spikes, they are the outermost molecules of the virus. And so they are antigens. The spikes also serve as attachment points for binding to host cells. So that this virus here, if I can get the mouse to work. Come on, mouse. There it goes. The molecules of the virus which attach to the host will be these molecules right here on the outer side of the spikes. The antigens and the attachment points are always the outermost molecules of the virus. So of a virus that doesn't have spikes, say a virus that's only a naked virus that only has a capsid, where are the antigens, as well as where are the attachment points for the molecules of the virus that bind to the host cell? Come on, I've already told you, it's even on the slide here. Okay, they would be the capsid because they are the outermost molecules of the virus. And the outermost molecules of the virus are always the antigens, and they are also always the molecules of the virus that bind to the host cell. Any questions about any of that? When we're talking about the viral structure, it's important to talk about the morphology, that is the shape of the viruses. Viruses are actually classified on the basis of their capsid structure. You can have a helical virus where the capsid is in a helix. I don't see it here, but Ebola is an envelope virus, and that sort of makes it hard to see the helix of the capsid. And then there are polyhedral viruses. These are viruses that look like a big dice. There's an actual polyhedral virus right there. That's an electron microscopic image of one. And then the third virus morphology is complex, where the virus can take on essentially any shape. Like there's a complex virus, here's a complex virus, and there's a complex virus there that looks like the lunar lander of the Apollo space missions. Any question about any of that? We'll talk a little bit more about this virus. It's bacteriophage T4. Bacteriophage T4 is a virus that infects bacteria. Why it's called the bacteriophage is it's a virus that infects bacteria cells. And T4 is one that infects E. coli. T4 is the virus that has been studied the most, more than any other virus. So we know the most about it. The reason why T4 was selected to study is T4 infects E. coli cells. An E. coli cell was a cell that was very easy to have, and most microbiology labs already had them E. coli cells in the lab. And so they studied T4 just because it infected E. coli. T4 has different capsomeres. We did mention that's the protein in the capsid. And there's generally one capsomere in the head, this portion of the virus here. The head also contains the nucleic acid, which you can sort of see there. But there will be a different capsomere, probably two different capsomeres in the sheath, a different capsomere in the base plate, probably a different capsomere in the pin, and then a different capsomere, different protein in the tail fiber. Any question about any of that? T4 belongs to the T. even family of bacteriophages. We give them names T2, T4, T6. They're all bacteriophages, meaning they infect bacteria cells. Furthermore, the adhesions, the molecules of the virus that bind to their host cell, are on the bottom of the tail fibers. So this region here and here is what is the viral molecule that binds to the host cell molecule. Any question about any of that? When a virus infects an animal, particularly a human, but any animal, the host immune system will respond. So a virus will get in an animal, it'll stimulate the animal to produce antibodies. Antibodies recognize and bind to viral surface protein. These are the antigens. And what the virus recognizes are the surface molecules, as well as it's what the antibodies will bind to. Some viruses can change or mutate frequently. So the different viral strains can therefore infect the same individual more than once. So even if you're immune to the virus could change, and then you can be infected by a different strain. Can anyone give me an example of that? Where the virus puts out new strains, and then you can become infected by the new strain.
Influenza virus.
Say again?
Influenza.
Oh yeah, influenza is a very good example. That virus changes more rapidly than any virus that I know. And actually, let me go ahead and talk about this now. And that's why you should get a flu shot at least once every 3 years. Let me see if I can get paint to come up. For some reason, this is not working the way it should. Let me see if I can do this. No, that's not what I want. Oh, there it goes. So when you have immunity, and this would be like 100% immunity up here, I'm just going to do this like that. Not very good, but you understand. And then down here would be 0% immunity. And this is just time on this axis. Let me see if I can write that. Up. Harder to do than you'd think. There we go. When you're first given the vaccine, and it's true of all people, the immunity will never be 100% because not everyone responds to the vaccine, but it's usually, especially with young people, pretty high, like 90 to 95%. And over time, that immunity decreases. After 3 years, the immunity drops below a certain threshold level where they say you are no longer protected. And we're talking about the flu shot here, by the way. So the flu vaccine. So after 3 years, you need to get another flu shot because the immunity is so weak at this point that you're not being protected. Now, obviously, the immune system decays with time, not the immune system, the immunity. And ever since you get the vaccine, I don't know, like the first 2 weeks afterwards, or maybe the first month afterward, the immunity starts to decrease. There is a time when the flu shot will drop down to very low numbers. I don't know when that is, but essentially zero. But after 3 years, it drops below the level that they say that you're protected. So they say with a flu shot, you have to get it at least every 3 years. And they recommend getting it every year so that your immune system, your immunity is high. Any question about any of that? The reason why this has to be done at least every 3 years, or maybe even every 1 year to keep the immunity high, is because the flu vaccine changes rapidly. Now, for something like smallpox or polio, that immunity for these viruses lasts, generally speaking, for your life. So I got my polio shot, which you guys probably didn't even get, when I was a small child. And smallpox also when I was also a small child, and I'm still immune. So these viruses, which were made in the 1950s or earlier, are still the vaccines that are being used today. So these viruses are not changing very rapidly. And it's a good thing that they're not, because otherwise you'd have to, remake the polio vaccine, and you'd have to go get a polio vaccine shot at least every 3 years, or not every year. Unfortunately, only with the flu shot, influenza, and then COVID-19, these viruses change very rapidly. And you will probably need to get a vaccine for these about once a year, at least once every 3 years. Any question about any of that? Hopefully I didn't shut that down. Oh, there it is. So some viruses can change or mutate frequently, and then you need to get a vaccine frequently because a different viral strain can evolve and then can infect you. And as you know, that's what's happening with COVID-19. The original vaccine, when was that made? In 2020, maybe 2021, something like that. No longer protects you against the viruses that are circulating today. Any question about any of that? All right. When we're talking about viral taxonomy, we can group viruses into families based upon different characteristics. You can, let me see if I can remember now. King, Peter, son, came over from. So we can group viruses into orders, but grouping them into a higher group than an order is, for most viruses, impossible to do. The reason being that viruses are a very small genome, and they evolve very quickly. They evolve quickly to their host. And so we just do not know the evolutionary history of viruses long term. So we group viruses into families and into orders, but above that, we don't group viruses into groups, with one exception. We do group them into the kingdom that they infect. Like there are animal viruses that infect animal cells, and there are bacteria viruses which infect bacteria cells. We don't know this from the evolutionary history of the virus. We just know it from the cell that the virus is infecting. So we do group them into the kingdom that they infect. And then we don't group them into subkingdom, phyla, whatever comes after that, maybe order. We do group them by families, by the type of nucleic acid they have, the replication strategy, and the morphology that the virus has. The family name of a virus ends in viridae. The genus name of a virus ends in virus. So for example, for the herpes family of virus, we call it herpes viridae. And then for the genus name of like herpes simplex virus, it's herpes virus. The genus name ends in virus. The viral species is a group of virus sharing the same genetic information and ecological niche, meaning they all infect the same host. And with viruses, we have subspecies, and these are given the species name and then designated by a number. Like with the herpes simplex species, we have herpes simplex type 1 and herpes simplex type 2. For the poliovirus, I'm trying to remember what the name of that is. Is it just called poliovirus or human poliovirus? It must be poliovirus. It is poliovirus type 1 and poliovirus type 2. You'll be pleased to know that viral species are not given a Latin name. They are given common English names. So wherever you are in the world, the poliovirus is called the poliovirus. Everyone uses the English name to name it. It turns out that the first people who were naming viruses, maybe the first virologists, were all English speaking. And they named the viruses. And then when the other people who spoke other languages started studying viruses, they just adopted the English names. And so viruses only have common English names. They don't have a Latin name to name them. Does that make you pleased? Anyone? I would think you'd be pleased that you only need to know the English name. The subspecies of a virus is extremely important. A subspecies, if you don't know, would be for humans, that would be the different races. Like the blacks are a race, and then the Europeans are a race. Well, for viruses, the subspecies are a little more important than the color of someone's skin. An example is human poliovirus 1 causes the disease polio. Human poliovirus 2 is the vaccine that we use in the oral poliovirus vaccine to prevent people from getting polio. So it tells you how different the subspecies can be of a virus. One subspecies causes polio, the other one we use as a vaccine to prevent getting the polio disease. Any question about any of that? Another example is one that you already know is different. Herpes simplex type 1 and herpes simplex type 2. Most people consider them different viruses, but they're the same species. They just infect generally different areas of your body, and the disease is different too. Subspecies type 1 is much less severe than subspecies type 2. So I've already given you an example of viral taxonomy. For the herpes virus, the family name is herpes viridae. The genus name ends with virus, so it's herpes simplex virus. The common name is herpes simplex virus, but the scientific name is human herpes virus. The scientific name has not been adopted, generally speaking, by even the medical profession. Most people do still call it herpes simplex virus. Okay, so this is one point where scientific name has not even been adopted. And then there's different subspecies like herpes simplex virus type 1. For growing viruses in the lab, it's actually very easy to grow viruses which infect bacteria cells in the lab. And that is all you do is you grow them in bacteria cells. I used to do this when I was an undergraduate. Did I ever do this? Yes, I did it as a graduate student as well, and as a postdoc. Viruses can be grown in the lab. What you do is you get your petri dish, and then you get the bacteria to grow everywhere on the plate. We call that a lawn of bacteria. That's why the plate, let me blow this up, is off-white in most places. That's because a lawn of bacteria is growing. And then you put the solution containing the virus down, and wherever a virus goes, it will infect a bacteria cell, replicating the virus. The virus will lyse the cell, be released from the cell, go out and infect more cells, and then you'll get eventually a plaque growing from where that virus initially infected the cell. Come on, mouse. Now, as you can see, let me blow this up a little bit. You can probably see that this is a merger of 3 plaques there. But that plaque is smaller than that plaque and that plaque. One reason why is probably initially when the viral solution was put down, the virus immediately bound to a bacteria cell and then infected the bacteria cell and replicated. Whereas this virus was near the bacteria cell, but the bacteria had to grow and probably replicated a few times before the bacteria cell got to where the virus was, and then the two touched each other, and then the bacteria cell became infected and became lysed, and then more viruses were replicated, and then that began the start of the plaque formation. Any question about any of that? All right, so in viral multiplication, the viral, excuse me. I knew I was gonna sneeze. That's why I grabbed my handkerchief. The viral genome only contains a few of the genes needed for new virion synthesis. How viruses multiply is they hijack their host cell. So the virus will bind to a cell, and if it's a host cell, a virus that can infect the cell and then use the cell to replicate in, it'll hijack the cell, take over the host cell's metabolic machinery, and then use that machinery of the host cell by using the host cell ribosomes, tRNA, amino acids, ATP, some enzymes, use this machinery to replicate more viruses. And the basic mechanism of viral multiplication is similar in all viruses. For bacteriophages or viruses which infect bacteria cells, and that's what we mean by bacteriophages, these are viruses which have 2 mechanisms of reproducing: the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, when the virus infects the host cell, it will replicate in the host cell and then lyse the bacteria cell, why it's called the lytic life cycle. And the bacteria cell is killed, meaning the host cell is killed. In the lysogenic life cycle, while the virus remains in the lysogenic life cycle, the host cell will remain alive. In fact, the virus, when it's in the lysogenic life cycle, will form symbiosis with the host cell, and the two will live together. Any question about any of that? Those are the only 2 ways that bacteria viruses can replicate. So to make new bacteriophages, what do we need? What does a virus need? What components need to be made in order for a virus to replicate? Come on, anyone? Well, you need to replicate the viral genome. And in this slide, we're only showing it as double-stranded DNA just to make it easy. That needs to be replicated, make new double-stranded DNA. The DNA has to be transcribed into viral messenger RNA, and then the viral messenger RNA has to be translated into the viral capsid proteins. And the capsid proteins need to get together with the newly replicated viral DNA to make the new progeny virions. This is the minimum that we need. Any question about any of that? In the viral lytic life cycle, the viruses multiply by going through 5 stages of the lytic life cycle. The first stage being attachment, the second stage being penetration, the third stage being biosynthesis, the fourth stage being maturation, and the fifth stage, release of the newly replicated viruses. Any question about any of that? All right, if there's no questions, let's watch a little video on the lytic life cycle.
First, the phage particles stick to the surface of the bacteria.
Oops, I didn't mean to stop the share. Didn't mean to shut this down, though. I need to convert that, and I don't have the software to doing that because this player can't read it. So let's start this one.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions about that movie? Let's go through the different 5 stages of virus attachment in the lytic life cycle. First, the virus has to attach to the host cell. How that happens is there are adhesion molecules on the virus which bind to molecules on the host cell. And some people call them adhesions, the molecules on the virus. Other people call them ligands. On the host cell, the molecules that the virus binds to are called receptor molecules. with the bacteriophage T4, like I mentioned, the bottom of the, let me blow this up. The bottom of the tail fiber is what binds to the host cell receptor. So first is attachment. 2 is penetration. That is where the virus injects its nucleic acid into the host cell. How that happens is the sheath actually contracts, and it is true, the little movie told you, it gets rid of the cell wall right there. And then the virus, when it contracts, it can put the virus through the plasma membrane, which would be the yellow thing right there. And when the sheath contracts, it puts pressure on the head, and that propels the nucleic acid, which is in the head, into the cell, much as when you push down on a syringe, it pushes what's in the head of the syringe into the patient. Not shown here. Let me see if it's on the next slide. No. Go back. One of the first things the virus does is it will digest the host chromosome. Why the virus digests the host chromosome is the viral genome wants to call the shots in the cell. And an easy way to do that is to digest the host chromosome, and then the cell can't use the host chromosome to carry out cellular activities. And then the viral genome is calling the shots in the cell. But what the viral genome does is it will be replicated to make new viral genome and transcribe to make newly replicated viral proteins, particularly the viral proteins of the capsid. The capsids then undergo biosynthesis, where the capsid molecules actually come together and form the virus, and the newly replicated DNA of the virus will be stuffed in the virus head, meaning there's an enzyme which actually stuff the DNA into the virus head. And then the virus self-replicate, meaning they self-assemble to form the new virions. And that's the biosynthesis stage of the lytic life cycle. Maturation is the assembly of the phage particles. And then there's the release stage. How that happens is a viral gene is transcribed, which digests the cell wall. And that will allow water to move into the bacteria cell. And eventually the cell will burst. Then when the cell bursts, that will release the newly replicated virions. Any question about any of that? All right, two terms you need to know: the burst time, that's the time from initial infection of the bacteria cell by the bacteriophage till the time when the cell will burst. And this will be between 20 and 40 minutes, depending on the species and the temperature that the virus is grown at. The burst size will be the number of newly replicated virions that will be released, usually someplace between 50 and 200 viruses. Any question about any of that? All right. That was the lytic life cycle. Now let's talk about the lysogenic life cycle. Upon attachment of a bacteriophage, which is a lysogenic virus, it will insert its genome, we're going to talk about it as DNA here, into the bacteria cell. One of two things will happen immediately, and that is the virus can decide to enter the lytic life cycle, in which case it'll go through the lytic life cycle that I just detailed on the lytic life cycle. So the lysogenic life cycle virus can enter the lytic life cycle. It can also enter the lysogenic life cycle if it's a lysogenic virus. If it decides to enter the lysogenic life cycle, the bacteriophage will remain inactive, and the bacterial host cell will remain unlysed, and we're going to call it a lysogenic cell because it's a bacterial cell containing a lysogenic virus, and the two will remain alive for as long as the virus remains in the lysogenic life cycle. Any question about any of that? All right, let's watch the little video further talking about the lysogenic life cycle. Where do I have it? Oh, here it is.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any question about any of that? So the lysogenic virus, once it infects a cell, it will inject its genome into the cell. We're going to talk about it as being DNA just to simplify things. The virus has to decide immediately whether to enter the lytic life cycle or the lysogenic life cycle. If it enters the lytic life cycle, the virus will be replicated, the genome will be replicated, viral protein will be made, new virions will be made, and then the virus will code for the destruction of the cell, releasing the newly replicated viruses. Okay, and that's the lytic life cycle, the same as we discussed previously. In the lysogenic life cycle, if the virus decides to enter the lysogenic life cycle, the viral genome will merge with the host cell chromosome. And when we have that, we call it a prophage. That is the virus genome merged into the host cell's chromosome. Where do I have that where I'm talking about? Oh, I got it next slide. Okay. So I'll talk about it later. And while the virus is in the lysogenic life cycle, the only time the virus replicates is when the host cell replicates. It'll replicate its DNA, and when it replicates its DNA, it's going to replicate the prophage, and then both daughter cells will receive a copy of that prophage. That can happen for many cell divisions. For reasons that are not fully understood, and they've studied this now for over 100 years, and they still don't have a good handle on it. for some reason, the lysogenic life cycle can discontinue and the lytic life cycle can start. What'll happen to start or to end the lysogenic life cycle and start the lytic life cycle is the prophage will be excised from the host cell chromosome. And then the lytic life cycle will begin, and that will kill the cell replicating the virus. Any question about any of that? All right. One reason why the lysogenic life cycle can continue is there is a repressor protein that is part of the lysogenic life cycle. And that when that repressor protein is active, it will bind to the DNA of the lytic life cycle and shut down those genes. So as long as the repressor protein is active, the lytic life cycle cannot begin. Any question about any of that? All right, there is one thing that they do know that can cause the lysogenic life cycle to end and the lytic life cycle to begin. And that is if you damage the host cell chromosome, the virus has some way of knowing that the host cell chromosome has been damaged, and the virus will excise and begin the lytic life cycle because the virus will want to replicate, and it knows that the host cell chromosome is damaged and that the host cell may not survive. But all other reasons for why or I should say most other reasons for why the lysogenic life cycle ends and the lytic life cycle begins, they haven't quite figured out yet. And like I said, they've been studying that for at least 100 years, and they just don't have a good handle on it. Any question about any of that? All right, there are some consequences of lysogeny. The lysogenic cell, meaning a bacteria cell that has a lysogenic virus in it, is immune to infection to other cells, to other viruses. So if there's another lysogenic virus, it cannot infect this cell that has a lysogenic virus inside it. So the cell is immune to infection by the same species of virus. And then the second consequence of lysogeny is the bacteria cell can be converted by the new DNA from the viral genome, meaning this cell here has gained new DNA. It's the viral DNA, and it becomes part of the host cell chromosome. And this can convert this cell to a new phenotype. This cell can express new properties of the virus. The best example of this is the Corynebacterium diphtherium. That bacteria only produces the diphtherium toxin when the Corynebacterium diphtherium bacteria is infected by a lysogenic virus. So the diphtherium toxin is a toxin on the DNA of the lysogenic virus. Any question about any of that? All right, that's enough talking about bacteriophages. Let's talk a little bit about the multiplication of animal viruses. The multiplication of animal viruses follows the same basic pattern of bacteriophage multiplication with a few differences. For one, attachment of the virus will be different because instead of attaching to the bacteria cell wall, you're going to attach to the cell membrane. Penetration is also different because it's much easier getting inside an animal cell that only has a cell membrane than it is getting inside a bacteria cell, which has a cell wall and a cell membrane. A penetration of animal cells happens by endocytosis or by fusion of the viral envelope to the cell membrane. The uncoating is similar. Biosynthesis is similar. Maturation is similar. And then release can be the same. We do have animal viruses which follow the lytic life cycle, where the animal virus actually ruptures the cell to release the newly replicated virions. But there are other ways that a virus can leave the cell, such as they can bud out of the cell, and this we call the budding life cycle. The budding life cycle only happens with envelope viruses. Any question about any of that? To my knowledge, if a virus has an envelope, it is released by the budding life cycle. If the virus has is a naked virus, at least an animal virus, it will be released by the lytic life cycle or a life cycle similar to the budding life cycle, which we're not exactly going to name. We're mainly going to talk about 2 life cycles in animal cells, well, 3 life cycles in animal cell, the lytic life cycle, the budding life cycle, and the, let me think, what's the third one? The latent life cycle. We'll talk about the latent life cycle later. Multiplication of animal viruses follows a similar pattern of bacteriophage multiplication with one major difference, and that is a bacteria virus has to replicate in the cytoplasm of a bacteria cell because there's no other place for the virus to replicate. However, for an animal virus, there is a big difference, and that is the animal virus may replicate in the cytoplasm, or it may replicate in the nucleus. Any question about any of that? Another difference with animal virus replication is the attachment. Viruses attach to the cell membrane. How they enter the cell is either by endocytosis or by fusion. If you have an older version of the textbook, it may say that the virus enters by penocytosis, and that is not correct. The newer versions and the latest version of the textbook have corrected that, and it does state by endocytosis. And the second way the virus may enter the cell is by fusion, where the viral envelope can fuse to the cell membrane and then enter the cell that way. Any questions about any of that? Well, I need to wash my glasses. Endocytosis happens by the virus attaching to the cell, and then the cell begins to vaginate around the virus, and then will be brought in further. This will pinch off right there, reforming the cell membrane, and then this will form a vesicle, which will bring in the virus into the cell. That's sort of showing the same thing. Let me show you fusion. Hopefully paint will come up I don't know why sometimes it just doesn't work. Oh, there it goes. All right, on a cell, we have a double membrane for the cell membrane. And for a virus, it will have a double membrane as well, meaning a lipid bilayer, which is a double membrane. When they touch, they can fuse Oops, I need to get the eraser back. Oh well. Just draw that in now. So the membrane forms the envelope with the virus when it fuses, and that will allow this virus to enter the cell. Any question about any of that? And that's by fusion. All right, I think I'm going to stop here before talking about the budding life cycle. So let's just state that the pattern of virus multiplication is similar to bacteriophage multiplication. There are a few differences. The main one being that the animal virus may replicate in the nucleus as well as in the cytoplasm. Any question about any of that? If not, I'm going to stop here and we will continue here next time. So if you have a question, show up for the lab at 6:30 or 6:30. If there's no questions, you do not need to show up in the lab. Any questions before I log out? All right, I'll see you next time.